Well, welcome to everybody who's joined us on this second session. And again, thanks to our supporters, without whom this wouldn't be possible. And as Jim just said, I didn't say anything about Potentilla reptans, Erecta and anglica and their hybrids in the first session because it's too much. So that's all I'm going to talk about today. Right, there was a bit of homework suggested. The point of that was simply to encourage you to look really carefully at these plants, because certainly Reptans and Erecta are so well known. They're the sort of things you don't look at carefully. You just say, oh yeah, there's Reptans, you know, Potentilla reptans, I know that, thankful. And you don't look closely. But you have to, certainly do have to when you've got hybrids involved. Right. Why do these cause so many problems? And they do. As I said last time, I've been BSBI referee for this genus for a long, long time. And at least 95% of my inquiries are about these three species and their hybrids. So there are problems. We're beginning, actually, I've got a bar up there. Can I get rid of it? No, I can't. No worry, sorry. We're beginning with Potentilla reptans, familiar species. But just to make the point, these are grassland species, and so they get all tangled up with grass and stuff. So to see them properly, really, you do have to sort them out, and herbarium specimens are actually the best way to look at these plants, and that's what we'll be doing. And all of the sheets that you'll see are my own. A few of them are of plants that were sent, specimens that were sent to me as referee, where the sender said, I'm happy for you to keep some of the material and use it for your own purposes. Right, a press specimen of Potentilla reptans, and you can see that it has, let me find the pointer down here. Oops. The laser pointer, I think, is quite nice. Okay, you can see that it has very long runners. The runners carry on growing all season. They don't stop till the weather stops them in the autumn, basically. Branching very occasionally, not often. 1 to several leaves at each node. So several there, 1, 2 there, maybe. All the leaves have 5 leaflets. I mean, okay, I have once or twice seen one with only 3, but that's just an aberration. You can ignore that. Petiole lengths generally quite long, but they vary, and they're not always long, as you'll see in the next slide. 1 flower per node, and that's quite regular. So here in the youngest node, a node is the point where the leaves arise, you've got a bud. Next one back, you've got the open flower. Next one back, you've got a flower that's over. And then here there's another slightly older one. And then here there's another one, and so on. So very regularly, 1 flower per node. All the flowers have 5 petals, not 4. And the runner can root, and it will be rooting at some nodes, you can see that here. But any of these nodes are capable of rooting. If you put them in a jar of water, they will root. So note, very important point, the leaflet size and the petiole length remain constant from node to node all along these runners. They don't get smaller as you go away from the parent plant. 2 specimens to show you the range of environmental variation in this species. And I talked about that in the first webinar. There is huge environmental variation in this genus, and you must take that into account. So don't bother about the size, but look more at the qualitative characters. So if we look at this little widdly one, and you can see there's the same scale I've put on all of the sheets, so that is 20 centimeters. These are at the same magnification. And here the leaves all have 5 leaflets. Cinquefoil really describes this species. And the flowers will all have 5 petals. Potentilla reptans is a tetraploid species. The base number for the genus is 7. So the chromosome number of Reptans 2n equals 28, which is 4x, 4 times 7. It's a tetraploid species, which is a very common number in this genus, actually. Now, Potentilla reptans is self-incompatible. That means that self-pollination will not result in seed set. The plant can block its own pollen. Now, a single clone of Reptans can cover a very wide area, a whole field, a whole roadside, whatever. So that means that plants often lack a compatible mate. In other words, another Potentilla reptans flower that's not from the same clone. So therefore, you very rarely see ripe seed on Potentilla reptans. I don't suppose you've ever looked for it, but when I did, I realized that. In that case, how can you judge the fertility of the plant? There's no seed. Is it fertile? Is it sterile? You don't know. The way you can tell is by looking at the pollen. And get that by squashing anthers, stamens, in a drop of stain, but you have to have a microscope. The stain doesn't have to be very technical. Red food colouring from the supermarket does perfectly well, but you have to have a microscope. And here you can see the pollen grains are more or less sort of triangular spherical. They stain a nice deep pink, and Reptans is almost 100% fertile. There is just one pollen grain up there, so the pollen count here will be nearly 100%. So the plant is fully fertile, even though usually it doesn't set seed because it hasn't got a mate. So now, moving on to Potentilla erecta, tourmentil, a very familiar plant, very widespread, growing on usually acid soil. Right, this one, the flowers obviously all have 4 petals, and that's the distinguishing feature of this. The stem leaves, the corline leaves, just have 3 leaflets, but we'll see that better in a minute on a pressed specimen. What's indicated here is a rather different looking leaf, and this is a basal leaf from another plant, obviously, that's growing just here. The basal leaves, of course, have stalks, and the leaflets are much wider. So this is another tetraploid species, 2n equals 28, 4x, the same chromosome number as Reptans. Specimens at the same magnification, both from lowland English sites. B is, looking at the scale here, it's just over 10 centimeters tall, and that's a typical size for Reptans, certainly as I see it, where I live. This one on the left is huge. It was from another lowland English site, but that's very, very large. But I've seen specimens much smaller than this. So again, we've got this huge size variation, and you just have to accept that. Don't worry about it, really. Stem leaves here always ternate, always 3 leaflets. You can see it very clearly there. And they are all sessile. In other words, they're unstalked. I mean, we can allow maybe 1 millimeter of stalk, but that's it. Stipules are large almost as the smallest leaflets, but they're a different shape. All the flowers have 4 petals, as you just saw. Now, let's have a look at some of the leaflets, though. These were just things I picked from my various pressed feeds. Very variable when you look at them closely. This one's narrow. It's got, the teeth are all in the terminal part of the leaf near the apex. The stipules are quite deeply toothed. This one rather similar, but the leaflets are broader. The teeth go down quite a bit nearer to the base. Stipules similar. This one, leaflet similar to that, but stipules are entire. They're not toothed at all. Similarly with this one, and this one's got much broader leaflets with rather shallower teeth all in the top half. And this one is really, really weird. I can say no more about that. It was in Cambridgeshire, so lowland England, growing in a fen. So whether that made a difference, the overall plant looked like Potentilla erecta. It was tall, But, you know, so there's huge variation in the leaflets of Potentilla erecta, and you must allow for that as well. However, Erecta has two described subspecies. There's the typical plants, which are subspecies Erecta, and there's a generally upland subspecies called subspecies Strictissima. And this has leaflets, and here's a specimen of it, and this bit is blown up over here. So the leaflets have got very deep teeth, and they go down quite close to the base of the leaflet. The leaflet's much more oblong, sort of, than usual. And the stipules are very deeply divided, so they're sort of spiky. Now, you can see the heading. There was a letter here, and you can see that it's too pale to see. But notice, University of Dublin. And the letter here explained the habitat, which was from a coastal marsh in County Wicklow. So this is definitely not an upland site. Now, all I can say here is I sometimes get specimens and people say, is this Strictissima? And I look at it and I think, I don't know. It's not from an upland site. The leaflets are somewhere between this and some of the ones I showed in the last slide. It's a big plant, it's a robust plant, like the one I showed you in the previous slide, you know, maybe 40 centimeters tall. I just don't know. And frankly, this needs some work doing on it, I think. Somebody needs to have a proper geographical look at these things, but not me. Erecta is also self-incompatible. There's the pollen, it's nearly 100% fertile, it's perfectly fertile. But Erecta, the small plants are erect, there is no vegetative propagation, and it normally grows in big populations of separate plants. So there's never any problem with cross-pollination there. Every plant always has lots of mates around it, basically. Moving on now to Potentilla anglica, trailing tourmental. This forms runners, but they're quite short. They will root, but they don't do it probably till late in the season. The leaves have 3, 4, or 5 leaflets, and looking around here, you probably see, well, mostly they seem to have 5. There's a 3. Flowers with 4 or 5 petals. This one obviously has 5, but 4s are probably more common. Now, this one is self-compatible. In other words, its own pollen can fertilize the ovules. And for this reason, you will always expect to find this plant setting seed. Even if you've only got one plant, it can fertilize. Sorry, pollination and fertilization are two completely different things, of course, but this can fertilize itself. So it regularly sets seed. It has a higher chromosome number than the other two, 56 chromosomes, which is 8x. So this is an octoploid species. 2 herbarium sheets, well, you can expect this again. Huge variation in size, of course. The great big one on the right is an Irish one. Ireland is a very good place for these species, I have to say. This one on the left, look at the leaflet number. There's 4, 5, 4, going further up. There's just about 4 there, and then there's 3. In most specimens, most of the leaves are ternate, but these two are perhaps not quite typical in that. This plant, at a certain time of the year, it really goes into flowering mode. So the runners don't go on sort of indefinitely, as the runners of Reptans do. They start branching more frequently, they have more flowers, the leaves get smaller, and it definitely goes into flowering mode. So over here, this one, just point out, the leaves are very large, but still this flower's only got 4 petals. So it is clearly, and it's got leaves with 3 leaflets, a lot of them. So it's clearly Potentilla anglica, not Reptans. The pollen of Potentilla anglica is slightly less fertile than that of the other two, and that's because it is a higher polyploid. It's got more chromosomes to manage, basically. But it's fertile enough to get always good seed set. And as I said, it will always fertilize itself. The pollen grains are larger than those of Erecta and Reptans, and that's because of the higher chromosome number. And I said in the first webinar that cell size is proportional to chromosome number. So if there are more chromosomes, you get bigger cells and bigger pollen grains. And if you look at these three, Erecta pollen grains are larger than Reptans ones. That's just a genetic difference between these species. But the anglica ones are larger than either. So for me, if you gave me a decent pollen slide, I could tell you straight away if I was looking at pollen of Erecta, Reptans, or anglica. It's a really, really useful feature, but you have to have a microscope. That's the snag. This is a picture I made a long time ago for a publication. Silhouettes taken from actual herbarium specimens. A is Reptans, 5 leaflets, flower with 5 petals. B is anglica, and C is Erecta. And you can see, just looking at these, anglica is intermediate in almost every character, leaflet number, petal number, everything. There are just a few differences. The first one is the leaflet width is broader in anglica relative to length. So look at that. I describe them as fan-shaped, really. They've got a wider basal angle, the individual leaflets. Looking at Reptans, they're much narrower, and Erecta, they're narrower still. Now, again, this is something I talked about in the first webinar, that polyploidy causes an increase in leaf breadth. Slightly strange, but it does, and I showed a very good example of it last time. So that's a polyploid character because it has 8 sets, not 4. Second thing is you get occasional small, simple leaves. And that thing arrowed up there is a leaf because it's got a stalk and it's got teeth. It's not a stipule. You don't get them in Reptans and Erecta, as far as I'm aware, but you do get them in anglica and in the hybrids. It's just a curiosity, basically. And the other important difference is it's self-fertile. Erecta and Reptans are not. They're self-incompatible. So, I mean, looking at this picture, it's obvious that anglica arose as a hybrid between Erecta and Reptans, and that is accepted. Hybrids are often vigorous plants. We talk about hybrid vigor, often applies to garden plants. The trouble is that they are almost always at least partially sterile, but many of them are completely sterile. And there's, I would say, almost always some degree of sterility. So unless they can propagate asexually, and in this case it would be runners or apomixis, they have no long-term future. They're just, hybrids are really just curiosities. Now, chromosome doubling provides an escape from sterility. This sounds horribly genetical. Take a deep breath, don't panic. This picture shows you some chromosomes of a fictitious plant, plant blue. It has a base number of 3 because I couldn't be bothered to draw anymore. And each set of chromosomes contains one big one like a wooden clothes peg, and one little one. And so that's one set, that's the other set. So the chromosome here, chromosome number is 2n equals 6. That is 2 sets of 3. One set will have come from the female parent and the other set from the male parent. When this plant forms eggs, egg cells, and pollen grains, the chromosome number will have to be halved because you need only one set in each gamete, egg or pollen grain. So this is the process of meiosis. If you've heard that word because you've studied biology, well and good. If you haven't, it doesn't matter at all. So the process of meiosis involves, first of all, and absolutely essentially, a pairing of the matching chromosomes. It's a precise molecular pairing process. If this doesn't happen, this division will fail completely. What happens then is that the partners separate. One goes to one end of the cell and the other goes to the other end of the cell, and you finish up with one perfect set at each end of the cell, and these are what go into the gametes, the eggs, and the pollen grains. Okay, that is meiosis. It's a complicated process. It's essential. Now, what happens in a hybrid? So now we have blue plant hybridizing with yellow plant. Yellow plant is a related species. Its chromosomes look the same, but actually the DNA in them will be different, so they don't match the blue ones precisely. The hybrid in the middle here has got 6 chromosomes, 3 from yellow and 3 from blue. And let's say it's got hybrid vigor. It's a lovely plant, very nice. But when it comes to meiosis, the chromosomes don't match precisely, so they can't pair. And the result is that they can't separate one to each end of the cell, and there's a kind of chaos. And that hybrid will therefore be sterile. So that is the basis of it. Now, this is your hybrid. It's got 3 yellow, 3 blue. Supposing the chromosome number is doubled, each chromosome now has a perfect matching partner. Meiosis will be completely regular. It's got twice the number of pairs that the parent plants had. That's fine. This becomes a fertile plant. And this hybrid is actually the complete sum of both parents. It's got all the chromosomes of both. And it's called an allopolyploid. That's a long word, but it is a really important word in plant evolution, because this plant will be fertile. It becomes a new species, basically, and it's a very important mechanism of plant speciation. It doesn't happen in animals, so zoologists won't learn about this, but for botanists, it's absolutely vital. Now we look at Potentillas, Erecta and Reptans, both 4x. Potentilla anglica is arisen by hybridization, but it's 8x. This is a classic allopolyploid. And you might say, well, why is it accepted as a new species? Why isn't it just called a hybrid? Well, because it's morphologically different from its parents. You can recognize it in the field. It's fertile, and it's reproductively isolated from them because it's got a different chromosome number. But its isolation is not complete because it does hybridize with Erecta and Reptans. And this is what causes the difficulties in the field, because you've now got a complete spectrum. So we start from pure Erecta, we recognize that. But then there are hybrids between Erecta and anglica, and they look intermediate. And then there's anglica itself. And then there are hybrids between anglica and Reptans, and they look intermediate. And then there's pure Reptans. And bearing in mind the huge environmental variation that you have in these plants, you can see the problem. It's very difficult in the field. So let's look at the hybrids. Sub-Erecta, that's anglica times Erecta, resembles Erecta, but it's probably a bit bigger, inflorescence a bit looser. The lower corline leaves may have 4 or 5 leaflets, and they are stalked. I haven't put that there, I should have. And some of the flowers may have 5 petals. The stems don't root, it doesn't have runners. Its habit is more like Erecta. And it has 42 chromosomes, it is 6x, because it's a cross between anglica, which is 8x, and Erecta, which is 4x, so it's 6x, 6 sevens are 42. A couple of specimens. And you can see in this one, particularly the lower corline leaf has 4 leaflets. That one's got 5, but then it reverts to 3, and there'll be 3 all the way up from there. And this one is quite well stalked. In this specimen, the stalks are much shorter, and all the leaves have only 3 leaflets. The leaflet shape is different. This was a completely different specimen from a different site. But even up here, there are short stalks. well, as I said before, Erecta is only allowed 1 millimetre of stalk length. The leaves should be sessile. So, yeah, that's Suberecta. Now, these plants do not form runners. They won't spread vegetatively. So if you've got that hybrid, and you've probably got a lot of Erecta around, there must be the second parent. There must be anglica lurking somewhere. And people often forget to look for that. So if you've got the hybrid, anglica is rather shy. It tends to hide under the hedge in the ditch. You know, there's never a lot of it. But if you think you've got hybrids, do look, because anglica may be a new record in that site. Who knows? Here is the pollen of Suberecta. It's partially fertile. It can be up to 40% fertile. This one is pretty sterile. But notice most of the grains here are unstained. They're empty. And those that are stained are variable in size, which should not be. And it's very easy to make this hybrid by artificial crosses. I did it with no trouble at all, with either Erecta or Reptans as the female parent. Sorry, Erecta or Anglica as the female parent. Just pollinate them with a paintbrush and you get seeds. It's easy. Second hybrid, known as Potentilla times mixta. This is Anglica times Reptans. This has the habit of Reptans. It has long rooting runners which go on running all through the season, cover a lot of ground. It has 5, 4, or 3 leaflets. You can see mostly 5s here. The flowers have 4 or 5 petals. So although this one's got mostly 5 leaflets, the flowers here, that's 4, that's 4. So the flowers with 4 petals are probably the most common. It is highly sterile, and like the other hybrid, it has 42 chromosomes. It's 6X, a cross between Reptans, which has 4X, and Anglica, which is 8X. 3 specimens. Well, again, size, hugely variable. Don't get carried away by the size, but count the leaflets. You'll find 5 or 4. Look at this little one, 5, 4, 3, and so on. Very long runners. Flower there. This one has 5 petals. So, long distance spread because of the runners. Now, okay, the story sounds quite simple to this point, and now it goes wrong, because Potentilla reptans does not hybridize readily, hardly at all, with Erecta or Anglica. So the origin of this hybrid must be a rare event. Now, I tried and tried with Reptans from different sites, crossing Reptans with Anglica, and I got no seeds at all. Well, none that germinated. I got seeds, they didn't, most of them were inviable. Someone in Manchester did manage to cross Reptans with Anglica. So it does happen occasionally. It probably depends on the genotype of the clone of Reptans that's involved. They sent me that plant, and I'll come back to that later. But you know, this hybrid obviously arises very rarely, but it spreads vegetatively and it occurs all over the place. So if you find it, you just call it Potentilla times mixta. Don't spend time looking for its parents because they may be hundreds of miles away. So this one then, we can call it a vegetative apomix. It's common, widely distributed, but don't expect to find its parents nearby. They do not cross at all easily. So, Potentilla mixta is completely sterile. None of its pollen takes up stain. It doesn't. So this is the whole set. Erecta has larger ones than mixta, both very fertile. Anglica has larger grains, very fertile. Suberecta has just a few fertile ones. Most of them are empty. Mixta has no stained grains at all. They're just all empty. So, I mean, as a referee, I just depend on this. Very often I find it really difficult to identify what I'm saying, and I look at the pollen, and it lasts. I've looked at, in one occasion, I had a sheet that was 100 years old, and I could still look at the pollen and it's still stained. So now the last possible hybrid is Erecta times Reptans, which has been called Potentilla times italica, and that must have 28 chromosomes, the same as both parents. But let's look at this bit from Stace, the flora. Potentilla italica basically is virtually impossible to distinguish from Potentilla mixta. That's what that is saying, other than by a chromosome count. Mixta has 42. Italica, if that is this hybrid, would have 28. To my knowledge, only one hybrid has ever been found with confirmed 28 chromosomes, and it's this. It turned up in Wales in 1983. It has 28 chromosomes. It's a rather odd-looking plant. Doesn't look hugely vigorous, but it's got great long runners. It's obviously a hybrid of some sort. This is the only known candidate for this natural hybrid Erecta times Reptans. Many crosses between these two species in Britain and elsewhere have produced only a handful of offspring, and I produced 2 using that Potentilla reptans that I was sent from Manchester. And one died within a few weeks. The other one was highly abnormal. I managed to keep it going just about into a second year, but it obviously was not a vigorous hybrid. So you are very unlikely to find that hybrid. If you find hybrids with long runners, it will be mixta. Let's go back a bit. Right, just to finish off then, this is the end of the story as far as fieldwork goes. Clearly, after all this work, I really, really wanted to recreate Potentilla anglica from its parents, because there's no question it is the allopolyploid between Erecta and Reptans. But I couldn't hybridize Erecta and Reptans and get a viable hybrid. Well, we assume probably that an allopolyploid arises from the hybrid, which then chromosome doubles, but it could go the other way around. So what I did was to induce polyploidy, higher polyploidy basically, in Erecta and Reptans, the parents, and try crossing them. And that is quite likely the way it arose in the first place, because from last week, you'll remember that polyploidy is rampant in the genus Potentilla. It happens naturally a lot. So I produced 8x Reptans, 8x and also 6x Erecta. The cross between 8x Reptans and 8x Erecta didn't work, but surprisingly, the cross between 8x Reptans and 6x Erecta did. And this, for me, was the eureka moment, because this cross, a cross between natural Erecta and Reptans, produced plant A, which is what you might call a synthetic hybrid. I don't like that term. We can't synthesize a plant. We just persuade the parents to cross. But B is a natural Potentilla anglica, and frankly, they are indistinguishable. The leaflet width, the number of leaflets, the number of petals, there's a 5 here and a 4 here. The little simple leaves, which there's some here. Not sure there's any in the natural one. And my synthetic hybrid, let's call it that, it only had 53 chromosomes, not 56, but that didn't seem to bother it. You know, you've lost 3, but you've still got 53. That's quite a lot. It was fully fertile and it was self-compatible. Everything just like the natural plant. And I was able to cross these plants with the natural anglica, and they were fully fertile. I was able to self-pollinate these, and they also produced proper fertile offspring. So that the last of the basic slides, this was the offspring of my synthetic anglica. So you see, it's fine, it's self-fertile, it's okay. So that was the end of my practical work with Potentillas, actually. I moved on to other things after that. But I'd just like to summarize all of this then, because I've said a lot, it's quite complicated. Looking at Potentillas, always allow for this massive environmental variation. Secondly, would somebody like to have a closer look at Potentilla erecta subspecies strictissima? I think it does still need more looking at. Thirdly, Potentilla times mixta is very common due to vegetative spread. It turns up all over the place, but don't try to look for its parents because it arises clearly very rarely and then it just spreads. Potentilla suberecta is quite different. This one doesn't spread vegetatively at all. It only occurs in the presence of both of its parents. If you think you've found it, look around, excuse me, for Potentilla anglica as well as Potentilla erecta, and you will find it somewhere. Potentilla anglica can be confused with mixta. Of course it can, it's difficult. The most important real diagnostic character is that Potentilla anglica will set seed, and Potentilla mixta times mixta never will. So if you find seeds, ripe seeds, that is, it's going to be anglica. Also, look at the leaflet breadth, although that's a more difficult, not such a easy character. The most unlikely to find the 4x erecta times reptans hybrid. It's obviously very rare. And if you're really keen, get a microscope. You can get a good but simple microscope. It's not that expensive. So that's it. Thank you.
Brenda, thank you very much. That was great.